Welcome to Learning Module 3. You have two PowerPoints here that cover what forensic context is, forensic significance, recovery of skeletal remains, postmortem interval, and then once you get to the lab, the process between initial examination and inventory. So in this module as well, you have finally been assigned your semester case project because now it's becoming relevant. So you are going to take your case, which is all just in Word form, literally in a Word document, and you're going to read through the relevant elements of that and connect it to these concepts that you're learning in this module. And this is what we're going to be doing for the rest of the semester with your case file and with the PowerPoints and readings. So get used to this. This will be a starter for you, so let me know if you have any questions about it. So we're going to start with this. What is forensic significance? So this is just, is this case worth looking at in a forensic setting? So the medical examiner's office, if it's an actual murder or something, should it be brought to court? And these are the things that the check boxes, if you will, that need to be met in order for a case to have forensic significance. So has to be within the last 50 years. The death needed to have occurred outside the care of a physician. And so, look at your case and see whether you think it fits those elements. This is not my favorite slide. It just looks at other ways for you to assess forensic significance. Color, texture, hydration, weight, condition, these all have to do with taphonomy. So taphonomy is like the natural processes that happen to something, in our case to human remains, after death, but it could be to an artifact after it's been abandoned or whatever. So it's a fairly blanket term. And here, basically, you're looking at decomposition, and to us of skeletal remains, right? Because we're not really working with fully fleshed remains very often, though we will talk about that a little bit here. So what's the color, right? Is it yellowish or ivory? Is it dark brown? If it's dark brown, it's very miscolored and it's likely to have been there for a very long time. If it's yellow and fairly clean, then it's probably relatively new. If it's greasy and heavy, then it's probably fairly new because it probably still has a lot of the oils in it that a person's bone has during life. If it's really dry and really light, then like lightweight, then it's probably older because it's probably very dried out. And the outer surface is probably a little bit pitted or even flaking, in which case it's older. So that doesn't mean over 50 years, it just means that it's been there for a while. So one of the things, the big picture things that you need to do with this material as we progress through here and through the reading is look at the postmortem interval. So the postmortem interval is the time that has passed since the individual died. This is really hard in forensic anthropology because you are working with skeletal remains. So what you essentially know is this person has been dead long enough that there's no more flesh. And you also need to look at those things like, is it still greasy? Is it yellow? Is it white and bleached and totally dry and super lightweight? Like this person would probably be. This person has been there a very long time, right? You can see that there's damage to the maxilla here, but overall, really, they're in pretty good shape. I mean, this is a thin area of bone. So there are some tables at the end of this PowerPoint, and there are also tables in your textbook, and you need to use those to determine the postmortem interval for your case. Okay? It shouldn't be something like this person has been dead for 5 years. It should be something like anywhere between 1 year and 15 years, or over 1 year. And this seems really counterintuitive to a lot of students because they think, I need to have a small postmortem interval. But it is more important that it be accurate than precise. So precise would mean narrow. Accurate just means you have the right time period in there. Because here's the thing, if you have misassessed your postmortem interval, if you say between 1 year and 2 years, and this person has been dead for 4 years, you look at a missing persons list when the time comes, then you are not going to find that person. So you have to have the correct time period in there. And with skeletal remains, that means that you tend to have to leave things pretty open-ended. So keep that in mind when you're working. We assess PMI very much through decomposition. So again, with soft tissue still present, it's considerably easier, but we look at the skeletal remains in and of themselves most of the time, or at least with very little soft tissue. So that said, let me warn you that the next couple of slides are the only slides where we really look at soft tissue in this class. And in large part because to understand what's going on to the body after death, we really should start right away. And so just warning, these are older pictures from the medical examiner's office that I think illustrate very clearly what we're talking about, but just so you are aware, these are probably the most graphic, I guess you could say, pictures that I show throughout the semester. It is always my goal not to gross students out or upset them or turn them green. I don't always succeed, but that is, I never want to turn a student green. So these pictures are the most graphic, I guess, that they really get. So you need to know these three things that happen after death. And most of these really start immediately because if you think about it, so one of the things we'll talk about later in the semester is cause of death and manner of death. The cause of death is ultimately always the heart stops beating. You're really asking what caused the heart to stop beating. But when the heart stops beating, gravity is the only thing acting on the blood. The blood is supposed to be bringing oxygen to all of your cells, and yet because you're not breathing and your heart's not beating, there is no more oxygen going to your cells, and so your cells are starting to die immediately because they need that oxygen and other things to process everything and to continue living, and so they start to die. And so all of this is a result of that. So rigor mortis, you have almost certainly heard of. This is muscle stiffening after death. And so at about 4 hours after death, you start to see this, and it will wear off after about 36 hours. So there's a window between 4 and 36 hours for rigor mortis, and it's to varying degrees. It sets in gradually and it wears off gradually. So this muscle stiffening, it can become so extreme that if you wanted to move the body, like move the arm or something, and the muscles, if they're in full rigor, you would need to really break the bone to move that arm. Now that's not done because you don't need to move arms in that way. But if you were not working for the medical examiner's office or whatever, maybe you were the killer, then you would have to do that. That's not a picture of rigor mortis. This is a picture of rigor mortis. You would need to break the arm or the leg. So here, this person died in this position and remained in this position for at least 4 hours, I would argue more, after death. And so you can see that though now they're actually laying on the floor, they're still in this position that they were originally in. And that will wear off again after 36 hours. Liver mortis or livor mortis. So it's supposed to be livor mortis, but nobody says livor mortis. And so one of my cultural pet peeves is, so now the people that say it right sound like they're wrong. So whatever you want to call it, I don't really care. But livor mortis, lividity, is when blood pools, because blood is no longer pumping through your body, gravity's acting on it. So both of these are pictures of livor mortis. Blood pools after death. It becomes visible at about an hour. It's fixed at about 12 hours, and at about 36 hours it disappears. So similar to rigor mortis. So if you overlap lividity and rigor mortis, then you may be able to estimate a tighter window in terms of PMI. Now again, we don't have all this soft tissue, so we don't tend to be assessing PMI with all of this. But just if you were, there you go. So lividity is kind of interesting. I'm sitting on a chair right now and I've got my feet propped up on a footstool. And it's kind of a hard footstool. And so they're angled downward, but they're above the floor. So the blood in my body, if I were to die in this position, would pool in the lower elements of my body. So it would pool on my butt because I'm sitting on a chair. But there's pressure exerted on my butt from that chair. And so what you see on this person's back is they were laying on a floor, and it kind of looks like a hardwood floor because you can kind of see lines through this. And so where there's pressure exerted from the floor onto their body, you don't actually see the blood pooled. So it would be pooled in my butt, but you would probably actually see like the fabric lines from my pants and potentially from this chair. It's got a kind of coarse fabric. Uh, you would see that in the blood pooling. So it would pool sort of in the areas where there wasn't direct pressure exerted. On my foot that's propped on this footstool, you would see this big white mark essentially, similar to this, because there's so much pressure being exerted by this footstool. But on the bottom of my calves, right, because my legs are sort of stretched out and angling slightly downward, you would have all of this blood pooled, and it would get to be a really dark color. And so the blood is just going to act with gravity, except when there's pressure exerted. As it starts to wear off, that's what you're seeing in this picture. So this is somebody at the ME's office wearing the purple gloves, and they've put their finger on the flesh here, and the blood moves away where they're exerting pressure. So this happens when lividity is starting to wear off. Algor mortis, then, is when the body is cooling. And so you can take the body's temperature, it will cool to ambient temperature. So if you're in a freezer, ambient temperature is below zero. If you're out in, the height of summer outside, it could be over 100. So in which case the body temperature would probably rise, right? So this is actually taken from the liver, which is annoying because the other one is called liver mortis. So sorry for that, not my decision. But so a temperature probe is actually put internally into the body, into the liver. And realize that immediately after death, because the body, the cells, etc., are going through this panic, is when I anthropomorphize them. And so the body temperature actually initially rises, and then it will cool to whatever ambient temperature is. And this is, of all of these, the least uniform, because ambient temperature is whatever it happens to be where this body is. So it's pretty highly variable. These are three things that you also need to know: autolysis, putrefaction, and then insect feeding. So really, these two are the ones that people get confused sometimes. So autolysis is exactly what it sounds like if you break that word down. It's self-digestion. So again, your cells in your body are dying. And your digestive fluid that's in your stomach in particular, it's contained by your stomach cells. And stomach cells are very, very fast replicating cells, and it is their job to contain that digestive fluid. Well, when they're not getting oxygen and everything that they need from your living body system, then they have to start to die too. And as they die, that digestive fluid leaks out and it starts digesting everything. So your own digestive fluid will start to digest your body, and it will start in the stomach region and sort of progress from there, so mainly in the abdomen. So this will start to happen basically immediately after death. It takes a while for it to move very far, but this does start to happen. Putrefaction, on the other hand, is when also having to do with your digestive system, but all of those little bacteria and microorganisms within your digestive system that usually work for you, well, they start working on you. So the bacteria start to eat the remains, and bacteria release gases as they metabolize, and so ammonia and nitrogen and carbon dioxide and methane, and this actually causes the abdomen to bloat. It also causes skin discoloration in the abdomen because of this buildup and all this bacteria in there. So putrefaction and autolysis are both about the breakdown of the body, but with autolysis, it's because of your own digestive fluids, and with putrefaction, it's because of the microorganisms that live in your body system. And then, of course, externally, insects are going to feed on the remains right away. I mean, if you think about taking a nap under a tree in the grass, you've got all sorts of bugs on you. So you're alive and you swat them off and whatever, but after death, that isn't happening anymore. So it's just a free-for-all. So that's also going to be part of decomposition. So all of these processes really go into decomp when it comes to human remains. So we'll move into the next installment of this with the rest of the slides.